final section of Chapter 1, the cell, involves the tools of cell biology, which includes different types of microscopy and a special technique called subcellular fractionation. So very simply, we'll talk about the different types of microscopy, whether it be from the light microscope or whether it be from the electron microscope, the pros and cons of each, and also some of the recent strides that light microscopy has taken to improve the resolution of different types of features found on a particular slide. And we'll also talk about the subcellular fractionation. So if I want to separate out ribosomes from the endoplasmic reticulum and from the mitochondria, I can do different techniques to isolate some particular types of organelles based on their density. When we talk about light microscopy, we always have to go back to the beginning with Mr. Robert Hooke, who coined the term cell after looking at pieces of cork underneath a microscope. This is further enhanced by Anton von Leeuwenhoek, who back in the 1670s under a light microscope also found that cells are in a particular shape after looking at different types of sperm, red blood cells, and bacteria. And then further on, back in 1838, Matthias Schleiden, who examined plant tissues, and Theodor Schwann, looking at animal tissues, also came to the same conclusion that all organisms are composed of different types of cells. So basically, we look at any kind of light microscopy or any kind of examining feature, whether it be something more contemporary, like looking at X-rays or looking at MRIs or CAT scans, or even looking at such things as our own glasses or contact lenses, you need what's called resolution. Very simply, being able to reveal fine details and be able to differentiate one structure for another. If a particular image or a particular apparatus like a microscope cannot differentiate one form from another, it doesn't give us a real good use to be able to detect one particular structure versus another, whether things look normal or whether things look abnormal. Figure 125 can give you an idea what the limits are of different types of microscopic evaluation. Light microscopy can give us at best maybe 0.2 micrometers. So you can see things up down to about the size of the peroxisomes and lysosomes, mitochondria, and then anything larger than that, including the upper portion of the cells. And of course, anything larger than that is easily seen by light microscopy. Anything smaller than a peroxisome or smaller than 100 nanometers will definitely need some help. We can still use some light microscopy with certain types of techniques. A lot of these are known as overlapping techniques, which we'll get to in a little bit. But at best, you'll be able to see mostly ribosomes and nothing really smaller than that. Trying to find any smaller structures, as far as like individual proteins or amino acids, you need some help with electron microscopy. Now, there are limitations with electron microscopy, which we'll get into later on. But as of yet, we do not have any good microscopy that can detect anything that's in the small molecular level. So an individual glucose molecule or an individual subunit, let's say a nucleic acid, cannot be seen due to the limits of even electron microscopy. Now, this is your standard compound light microscope with the light source found underneath the specimen, underneath the stage. The light will shine the wavelengths of light into the condenser and have it into a cone that will pass through the specimen, and whereas the reflective angles will actually be received into the objective lens. So by keeping that alpha angle tight as possible will give us the best resolution, but we're still going to be limited by the wavelength of light. So we're only down to about 1.4 possible value of resolution without any kind of additional help. So that's due to the medium of air as we look at the specimen. Now, that works well for our scanning as well as low and high magnification oculars. But we can actually do what's called oil immersion, which on some microscopes have a fourth objective lens. But the problem you run into is the oil has to be continuous from the specimen up to the objective lens. Any portion of air can reflect the light even more so, and then ruin the resolution of that particular image. So when we look at blood cells in particular in the lab, typically getting into high magnification, we cannot get enough resolution to look at one red blood cell from another, but adding a drop of the immersion oil can actually decrease that angle quite a bit and give us a better summation of the resolution of two different types of red blood cells or even white blood cell. But that little bit of oil immersion does help quite a bit. But again, that oil medium has to be continuous from the specimen to the objective lens in order to get the best resolution. Now, the most common type, or the one you're most familiar with in the lab, is what's known as bright field microscopy. So these use the compound light microscopes we looked at before, and then prepared slides to look at. Now, most of the slides have to be prepared in a certain way, which typically we take a specimen, whether it be, let's say, an organ or some piece of tissue, and it's usually going to be embedded in some kind of paraffin wax or possibly dipped in alcohol to preserve the structures, which unfortunately kills the actual tissue itself, but it preserves all the structures therein. Then they're stained usually with which is known as either H&E, which shows different types of stains for the nuclei. In this case, we're looking at nuclei as purple, the rest of the protein stained as pink. So this is actually a slide of a benign kidney tumor. So without these types of stainings, putting the light straight through the specimen, since the specimen is very thin, it'll pass it right through and then it'll wash out any kind of image you might see. So we stain these in certain types of slides we're looking for certain structures to kind of give us better resolution of those particular structures. Other types of stains include methylene blue, which kind of looks more at glucose-filled portions of the cell. There's other types of stains too help differentiate different types of proteins. The most common one, again, is H&E, which you mostly see the pink background in most types of tissues, especially mammalian tissue, human tissue. It gives a great look at what those cells actually look like. Now, if we want to look at live cells, there are techniques to use, in particular for light microscopy. One is known as phase contrast microscopy, found on the left, and differential interference contrast microscopy on the right. We'll go into further detail on each one, but very simply, we can take a look at cells that are live. In this case, these are epithelial cheek cells from the human mouth. You can get it from a cotton swab and just place it, swirl it onto a microscope and put a color slip on, and you can actually put it under a special microscope to get both of these types of images very easily. So first, let's look at the phase contrast microscopy. So very simply, use different types of optical systems to look at different parts of the cell. So what happens is with the nucleus, the light passes through the nucleus very poorly because of the density. So it shows up darker in this particular image, whereas it passes through very easily through the cytoplasm. So therefore, it's going to show more of a clearer picture here. But these are unstained slides, and these are live cells. So you can see them interact with different types of apparatus. Or if you want to inject, let's say, different types of fluids like saline or sugar solutions, you can actually see how the cell reacts to different types of environments. Or you want to change the pH, you can get a good idea of how things happen in real time. This is a differential interference contrast microscopy, which is a different technique. So this gives you more so, not so much shades of gray, but more or less just more density. It looks a little more three-dimensional than what you saw back on the phase contrast microscopy. Now, instead of using dyes, we can use a special substance called a fluorophore to give us what's known as fluorescence microscopy. So very simply, we use the different fluorophores to basically attach different structures within the cell, whether it be DNA or mitochondria, different types of organelles. And then very simply, when we excite these fluorophores, basically the electrons will be excited by the light energy, and then they'll move to more of an excited state. When they go back to that ground state, it actually releases other energy, in this case in the form of light, using a lower wavelength of what the original wavelength was that excited it in the first place. So that can bounce back different colors of fluorophores back to the eyepiece, whether it be ultraviolet, whether it be blue, whether it be green, red, depending on the fluorophore that we use, can help us determine different types of structures found within the cell. The most common ones are going to be the diamidinophenolindole and the HESH are going to be the commonly used examples. Both of them use bindings to double-stranded DNA and have an excitation wavelength of either ultraviolet or oftentimes blue for those particular structures. This also has another application if you're looking at immunofluorescence. If you're looking at antibodies, they can be tagged very easily, usually by a fluorophore known as fluorescein isothiocyanate, which will bounce back as blue or possibly even green. So these types of antibodies can be used to visualize a protein's localization in a particular part of the cell. In certain cases, let's say if it's a nerve cell, let's say if there's more proteins in the axon versus the dendrites, or a certain type of protein found in a certain type of muscle, whether it be skeletal versus, say, smooth, then we can tag those particular proteins using this type of microscopy. Now, you've seen most of these composite pictures in most of your biology textbooks. Really made things a little more colorful, a little more aesthetically pleasing to the eye. So again, very simply, what they're doing is they're attaching dyes to these particular structures within the cell. They're bouncing light or even ultraviolet light through what's known through a dichroic mirror to excite the electrons in a particular specimen. and when the excited electrons go back to the ground state, they emit a different wavelength of light up through the eyepiece. So very simply, you're seeing the blue upper portion here in the A area. That's going to bind to the actual nuclei themselves. You've got the green dye that's going to bind to the mitochondria, and you got the red dye is going to bind to different types of the cytoskeleton, whether it be microtubules, intermediate filaments, or microfilaments. And then the composite pick is going to be letter D. So you really see a nice spread of all three colors, and you get a real nice differentiation of each particular structure of this particular cell. Now, under fluorescence microscopy, unfortunately, the specimen has to be treated. It's usually therefore killed, so therefore it's not living cells. However, there's a newer technique that's been out for quite a while known as the green fluorescent protein. This is a protein that's derived from jellyfish, helps visualize proteins in living cells. So in this case, this was actually attached to different types of proteins along the mitotic spindle. And although small, you can get an idea of what's happening here. You can see in real time. You can actually see these in some videos posted on YouTube of different types of mitopically dividing cells with the green fluorescent protein attached to those particular proteins on the mitotic spindle. So you can actually see the cells in real time move from different stages of mitotic division. As we know, interphase is the time between mitotic divisions. So you can see very simply the green fluorescence is actually more compact. You see it starts to stretch out as you go through prophase, metaphase, anaphase, and telophase. You can see the differentiation of two separate daughter molecules almost in the anaphase, and you see both sets of chromosomes being pulled apart just before it gets to the telophase section. So while we don't see the cytokinesis, you can see almost the end result at the anaphase stage, where you see the two sets of chromosomes being pulled to the mitotic poles. Now, even with the benefit of using fluorescence microscopy, oftentimes the images can still be blurred. So you can use a different type of microscopy known as confocal microscopy, which gives you a better focus on the particular specimen at a better depth of the light that's being emitted. In this case, they usually a pinpoint of light, usually by a laser, which is going to be emitted into the specimen and then reflected back through what's known as the confocal aperture, which sharpens the image even more so because it allows only the light that's in a particular form of focus for that particular image, and basically blocks off any kind of light that might be out of focus. So this gives somewhat of a sharper image with the confocal microscopy, a little bit better than, say, a standard fluorescence microscopy. So here you can see the end result of not only fluorescence microscopy, but also the confocal microscopy as well. Notice how sharp the images are that we see through here. You see the nucleus in red, you see the actin filaments are in blue, and the microtubules found in yellow. You can see, obviously, it's a bigger image than the last slide we just looked at too, but only having the light coming off a certain position of the focus can give you a better resolution on all the structures found therein, and very crisp, very easily seen, and very easily differentiated. And we go even further into fluorescence microscopy by going into what's known as multiphoton excitation microscopy. So this one very simply is used for certain types of specimens, or looking for certain types of structures that are relatively thick, where a lot of times you can't establish a thin specimen to look at different types of structures, different cells. But if you can achieve a particular depth, or you have an idea where that particular specimen is, you use what's known as the multiphoton excitation microscopy. So very simply, instead of using one laser or one emission source, use two lasers that will very simply converge on one particular point, which will excite that particular fluorescent dye and give you a better image for that particular depth. So this is very easily used in living cells again, but it gives you more of a pinpoint structure, or more of a pinpoint idea of what the structures will look like by hitting only those structures. So therefore, you can actually look at this type of image over and over again very easily without damaging the particular specimen. As we know, even with long exposure to light, even some specimens will start to break down as the photon energy from that particular wavelength, that particular light can actually damage the specimen over time. Here we see 3 slides using multiphoton microscopy of mice brain tissue, detecting amyloid plaques, which is the benchmark for Alzheimer's disease. So you see they're stained in green, and you see the corresponding vasculature all in red. So you can almost envision a 3-dimensional structure by using this particular type of microscopy by different levels back and forth. different depths to give you more of a three-dimensional image of all three time-based slides here. So you can see the amyloid plaques really start to build up quite a bit by the time you hit it from week 8 all the way up to week 24. So this is a great way to see the cells as they're living, so you can get a quick snapshot of what's going on in real time. The next section of microscopy is known as super resolution microscopy. This always reminds me of the old TV shows where they always talk about taking pictures and they try and look closer and closer and enhance. But as we know, pretty much increased magnification does not always increase resolution. So if you look at the top portion here, all the conventional ones, they take a 3 micrometer shot and take that subsection and try and magnify. And unfortunately, there's not much improvement on the images, so there's no real improvement on resolution. Now, until a couple of years ago, back in 2014, there are 3 scientists, Dr. Eric Betzig, Stefan Hell, and William Murner, that actually got the Nobel Peace Prize in 2014 for chemistry for developing the super resolution microscopy. And that's what you see on the bottom here. That's nicknamed STORM, which very simply means stochastic optical reconstruction microscopy. So what this does very simply is, instead of taking one particular shot, it takes multiple shots of individual fluorescent molecules that are going to be embedded onto this particular area of the microtubules. So what it does very simply is it'll take a multitude of shots and then create a composite photograph, which you see all the way on the right-hand side under STORM, under 500 nanometers. So this is a composite of each individual molecule of fluorescence that is going to be shown on the image instead of all of them at once. So it's kind of like if you think about the lights on a string of Christmas lights, where they're either all on at the same time, or they might go on and off in sequential order. So in this case, it basically gives you less light pollution, so to speak, when you're looking at the last image there. So it actually increases the resolution by almost dimming down the rest of them on each individual shot, each individual image, and makes the whole composite, so it gives a nice little photo right through there. We move out of light microscopy and now get into what's known as electron micrographs, or electron microscopy, of which there's 2 main types. There's transmission and there's scanning. The first one we're looking at here is known as transmission electron microscopy. So this is basically a positively stained slide. So what they do is they will basically bathe the particular specimen in different types of heavy metals, like osmium tetroxide, uranyl acetate, and lead citrate, to basically stain different parts of the cell. And basically what happens is electrons are basically shot through the actual specimen here, and then if it deposits itself onto the film thereafter, it turns white. Any area that does not get stained remains black, so there's no reaction there. So basically it shows the more dense structures to be more opaque or more black here, and the more translucent or the thinner portions will be shown up closer to a white image. So this is kind of opposite what we would see, let's say, with an X-ray. So we deal with X-rays in my office quite often. So what we basically look for, anything that's very opaque, like bone, we show up as white, because very simply the X-rays get absorbed by the bone instead of being transmitted to the film behind it. So we see the opposite effect that we see here on this picture of the cell through a transmission electron microscope. So very simply, the electrons will pass through, so therefore it will show as a lighter type of part of the cell, whereas if it doesn't pass through, meaning it's absorbed by the specimen, then it'll show up as black. So this gives a nice gradient or grayscale, so to speak, which helps us differentiate one adjacent structure from another. Now this is also a transmission electron micrograph, but this is known as used in the technique known as negative staining. So these are actin filaments. So these are basically laid onto a heavy metal stain on the supporting film first. So that creates a dark background for the actin filaments to lay on top of. They are going to be surrounded by a film of electrodense stains. This gives you more of a light structure on a stained background. So this is known as a negative stain as opposed to the positive stain we saw on the previous slide. Now this is the second type of electron microscopy known as scanning electron microscopy. Now this one is a little bit different, where the electrons that are sent through don't necessarily pass through the actual specimen, but actually they're coated in some type of metal, usually gold. So naturally, this is done in a vacuum, just like the other transmission electron microscopy. So these tissues are effectively dead. So they will coat these particular specimens in gold or, like I said, another type of substance. And what happens is the electrons will bounce off the particular specimen here, and then they'll be detected on different types of apparatus to generate a three-dimensional image. Now, these can be colorized after the fact to differentiate different parts of the cell or different areas that they're looking for. But I always enjoy these because you can actually see some nice shots of phagocytic cells engulfing bacteria. You can see different types of actin and myosin filaments found deep within muscle cells or a neuron that forms the neuromuscular junction with the actual individual muscle fiber. It really gives you a good idea of the microscopic dimensional anatomy of different types of cells. It really gives a nice clear picture well, not real time, but basically you can really kind of see exactly what the structures look like, very dissimilar to what you see in the textbook as a conceptualized look at certain types of cell or certain types of structure. So we talked about looking at different parts of the cell, but let's talk about actually trying to separate different portions of the cell for further study. So we do a technique known as subcellular fractionation, which uses centrifuges. Basically what we do is a differential centrifugation, which basically takes into account the different densities of different portions of the cell. So as you can see here, basically what we're trying to do is we want to break down the cell at a point where it doesn't damage the organelles found within. So very simply, we pour the concoction of the cells into a centrifuge tube. We'll spin about 800 times gravity, about 10 minutes. That'll form a pellet on the bottom, which will have all the different parts of the broken plasma membrane, but yet the rest of the organelles will be intact. We'll transfer that into the next centrifuge slide, spin it at a faster rate. So that'll give us both a combination of the supernatant, which is the actual fluid left over, as well as the sediment that's going to be found deep within the area of the sediment that's going to be found within the centrifuge when we spin at a certain speed. So then now we can take out the different nuclei, which are going to be the most dense you're going to find because of all the genome, the DNA found within each nuclei. So once that's taken out, now we're left with some of the mid-range organelles. So we'll spin that at an even faster rate, about 100,000 times gravity, under what's known as an ultracentrifuge. So we'll repeat the process over and over again until we get down to the smaller and smaller parts of individual cells. So first we break down the cell itself at a speed that doesn't damage the organelles. Then we break down to the nuclei to try and separate those. Then next down to the mitochondria, the lysosomes and peroxisomes, and then into the actual fragments of the plasma membrane and the endoplasmic reticulum, and finally down into the ribosomes. And once we get down to the ribosomes, the remaining solution, the supernatant, the cytosol, that's the only left portion that's usable at that point. We simply separated not only the plasma membrane, all the portions therein, the different membrane fragments, and then also nuclei, then all the different organelles, then all the fragments of the plasma membrane and the ER, and then finally with the ribosomes. So now you're left with the supernatant of just the cytosol. Now we can separate those particular organelles even further by doing what's known as velocity centrifugation. So very simply, the specimen taken from the differential centrifugation will be placed through a sucrose gradient and basically let everything kind of settle through the particular sucrose gradient. So basically have a lot of fast settling particles, usually the smaller types of organelles like ribosomes will actually sift through first, whereas some of the larger ones like the Golgi apparatus, the R, and the nucleus, or even lysosomes and such, will actually band together as more discrete bands of different sizes that do not pass through the gradient very easily. And as you can see very simply, if you want to actually take apart the different types of sediments, you simply just puncture the centrifuge tube and just let it drop through until you get down to the next layer of the organelles very simply. It takes a little bit of time, a little bit of process, but it's an easy way to separate the ribosomes from the mitochondria from lysosomes, and as far as like the Golgi apparatus and the other organelles found deep within the cell. And finally, the last type of centrifugation, or trying to separate different types of organelles, is known as equilibrium centrifugation. So instead of letting it pass through a sucrose gradient, basically you can check different types of organelles based on their buoyancy. So very simply, they are centrifuged into what's known as equilibrium position, usually in a sucrose or what's known as a cesium chloride solution. And you'll see different bands that will basically indicate whether Like certain types of vesicles will be derived from either a rough endoplasmic reticulum or a smooth endoplasmic reticulum. They can be separated by using a micropipette very easily. So it can be involved in trying to check the synthesis of different types of proteins and lipids. As you know, the rough endoplasmic reticulum is going to be covered in ribosomes, so therefore the vesicles that pinch off from them should also have ribosomes there as well. So their sense of equilibrium, their sense of buoyancy may be quite a bit different from the smooth vesicles that come from the smooth ER. Now, as we wrap up chapter 1, this will be a quick little taste test of the actual assessment that's found at the end of each chapter. Some of the quizzes I'll be giving you, you will be graded upon, which when I understand in through Insight, they'll actually grade them for us, and I'll get the grades through Canvas through a link that I'll have later on. But looking at this particular problem, this gives us almost like a real-life idea of how these particular techniques, in particular fluorescence microscopy, is used to identify different types of partitions in the cell. So there's going to be two different sets of problems here. We're going to go to the next slide here. So looking at the top row, so very simply remember the DAPI is used to stain the nucleus of most of the cells here. Histones, if you remember, histones are going to be the proteins where the chromosomes will be wrapped around. They'll be densely packed deep within the nucleus itself. So if you look at both the DAPI and also the histones staining, the easy thing for you to determine is the histones are going to be found only, if not primarily, found within the nucleus itself. We look at the merged image on the far right. You can see that the green and blue is going to be superimposed on each other. The alpha tubulin fluorescence, if you notice, is going to be concentrated all around the cell, so it's going to adhere to the microtubules that are going to be found mostly outside of the nuclear area, so all throughout the cytoplasm up against the plasma membrane. So knowing what each one of these particular stains are going to adhere to can help us identify where in a particular cell we would most likely find those particular structures. The next slide here, so these four here, the main difference here is going to be known as the N-cadherin, and I'll give you a little synopsis of what that does. Cadherin is going to be a special protein that is going to adhere one cell to another. So this basically gives you inherent strength of that particular tissue. So we see on the far left, again, we see the DAPI is used to stain the nucleus of the cells, which you knew from the last set of slides. We find the N-cadherin is going to be found everywhere outside the nucleus, in fact, more so on the periphery, so the outer edge of the plasma membrane, as it's going to attach to the next most adjacent cell. So these cadherin proteins are more of structural proteins that will basically form a junction with one cell to the next. And again, we see the beta tubulin, which is going to be another stain used for the microtubules found in the cytoskeleton. So you'll find those usually found therein the N-cadherin stain, but also outside of nuclear stain. So if you look at the merged section on the far right, you can see all three colors are all merged together. So again, you can see the nucleus in the center of most of the cells, surrounded by the microtubules, and then finally on the vast edges, you'll find the N-cadherin through there. So I'll try and at least give you an example of each particular type of problem that you find on the actual quizzes. Some are just pretty much just trying to extrapolate from the information you learn from the sections of the chapter. I'll do my best. If there are some questions that are somewhat ambiguous, I will go back and hand grade some of them if they are a little bit tough on some of the material. Again, any questions on chapter 1, please contact me through my email address. If any of the videos you find me talking too fast or unintelligibly, please let me know as soon as possible so I can redo them. All right, thanks for your attention. Good luck studying on chapter 1, and we'll get started with chapter 2 sometime next week.